everybody, and welcome back to Biology 229 at HCC, Microbiology, Chapters 4 and 5. I am your host, Professor Mary Annarella, and we are working on Chapters 4 and 5. Chapter 4 is all about microbial growth, and Chapter 5 is all about the control of microbial growth. I just remembered that I did not turn on Do Not Disturb. There we go. Are we still recording? Yes, I sure hope we still are recording, because I already recorded this once and accidentally turned the microphone off, apparently, even though I thought it was on. So we're going to try this again, and we're going to try and do this without getting text from my mom interrupting us. Anywho, the PowerPoint for Chapters 4 and 5 is up on Moodle. And let's go through this. This is going to be a very PowerPoint-driven kind of thing. You'll probably hear a fan on me because it's pretty warm in my house. It's been a hot day. It's been a weird weather day. And I'm trying to stay cool without having to turn on the air conditioner. So microbial growth, we are talking about an increase in numbers of bacterial cells, not a small cell getting larger. So that's not what we mean by microbial growth. By microbial growth, we mean multiplication. So it's an increase in cell numbers, not cell size. And the way that bacterial cells increase their numbers is through a process called binary fission, which is a type of cell division. It is different from mitosis. I did it again. My microphone is on, and I was still in portrait mode. Here we are in landscape. Hallelujah, I got this in the first 1 minute. Anywho, we're going to be talking about doubling time on occasion, or you'll come across the term doubling time, which is the time for the number of bacterial cells to double. Doubling time is going to be different for different bacterial species. So it's not like, so if you ask me, well, how long does it take for bacteria to double their numbers? Well, what are we talking about? Are we talking about E. coli? That's about 20 minutes. For other cells, it takes longer. It also depends on the amount of available nutrients for them, and we'll be talking about that. But first, binary fission. Binary fission is not mitosis. It's kind of similar to mitosis in that everything gets divvied up inside the cell, but there's no nuclear division. There are no spindle fibers that form because prokaryotes don't have a cytoskeleton. So you don't, get a spindle forming at all. But what you do get is a bit of a cleavage furrow. So the DNA duplicates itself, and you get this cleavage furrow. Everything inside the cell gets divvied up. So binary fission is a pretty simple process compared to mitosis. All right, we've also talked in lab anyway about obtaining a pure culture. Why? Well, because in the environment you're not going to find just E. coli living all by itself. There's going to be a mix of different bacterial species in any environmental sample. So we want to obtain a pure culture, which I have up here, population of bacteria descended from a single cell, and the way we obtain that is through a streak plate. So you've already done your streak plates in lab, so you know that it works as a dilution. And there is an assumption, let me take a screenshot here for you, that what you're doing is that by the time, okay, this is quadrant 1, and we've got quadrant 2 here, looks like 3 and 4, that by the time you get out here, you will have diluted the cell culture out so much that you've managed to separate bacterial cells far enough so that as they undergo binary fission, they're going to increase in numbers and form a little colony like that. So the assumption is that one colony came from one single cell. The truth of it is that, two cells could be close enough to each other that they eventually start both undergoing binary fission and just kind of make one colony together, and you really can't tell. Like over here in the picture, I can't tell that this one colony right there is from one cell, or maybe two that were really close. You never know. There we go. So it is a bit of an assumption. Okay, and it requires a dilution. You've already studied that in lab, so I'm not going to go through that right here. But you do need to know the basics of a streak plate for lecture as well, because it's how we isolate a pure culture. All right, I'm not really crazy about this slide here. I'm not gonna talk about growth in a closed system. So you can go ahead and cross that out. Actually, let's change the color, cross that out. Continuous culture, we don't do that in the lab much at all, but we do compare growth on a solid medium versus a liquid medium, aka broth. On an agar plate, colonies are going to look different from each other. Some will be large and milky color, and some will be pinpoint. Some will have a very flat appearance, and some will have a very domed appearance if you look at them from the side. And in liquid media, if we have, various liquid cultures here, I know these are really crappy looking test tubes here, but some will give you bacterial growth that is all throughout. Some of it will kind of sediment, and that's just typical of the species. Some will float to the top. Some will just look very grainy throughout, and it's called a flocculum. But there are different growth habits there, and they will all be affected by environmental conditions as well. So let's talk about those environmental factors that affect growth. A huge one is temperature. Huge. Okay, so temperature. The temperature of a bacterium's environment will affect how well they grow. Oh, and I hear my dog clicking around again. I'm going to have to pause this for a second. Okay. And we're back. My dog definitely did have to go out. I heard that, I don't know, when you live with a dog or any pet long enough, you just kind of know their behavior. And I heard that characteristic click of her toenails on the floor and just how she was pacing, and I knew she had to go out. So, sorry. We're back now. We're talking about temperature. Temperature. Every organism, including humans, have their own internal optimal temperature. And for humans, it's around 37 degrees C, right? Which is, basal body temperature. That's just the temperature at which our cellular parts, especially our cellular proteins, work best. So likewise, bacterial cells will have their own optimal temperature, and the mesophiles are the ones that we are generally the most concerned about, because most human pathogens like our temperature. So it's mesophiles that are generally infectious to humans. There are thermophiles, which like it warmer. Some of them like that. And then hyperthermophiles really like it hot. So for example, within this range, for example, there would be Legionella, which is a microbe that causes Legionnaire's disease. It tends to like things nice and warm, such as, water that collects on a condenser for an air conditioner. An air conditioner has a fan, and so it's removing heat, and so you get some condensation on that, and that's where Legionella likes to hang out. Okay, and then mesophiles, and then those that like a little cooler, like cycrotrophs. Some staph species, some staph species like a little cooler. That's why they tend to infect human extremities sometimes, because people's hands and feet tend to be a little cooler than the rest of you. And then there are psychrophiles, which love it cold. Right, we actually use temperature as a means by which to control the growth of bacteria, but usually, you can boil things. You can use a high temperature to kill a lot of bacterial cells, not endospores. But if we're talking using cold temperatures, we only slow them down. Cold temperatures generally don't kill bacteria. In fact, we use the lyophilization process to store bacteria. We store bacteria in the freezer, and we pull them out and we can grow them again. You got to add a little bit of glycerin to make sure that we pull the water out of them, but it works really well. Cool temperatures only seem to slow them down. So there we go. So temperature will affect that. I just want to basically show you that take a screenshot first. That for food storage, this is just quickly to show you that a shallow dish here, I'm going to change my color right here. A shallow dish like this will cool very rapidly if it's hot and then you put it into the refrigerator. Whereas a deep dish takes longer to cool down to, 0, to freezing. So this isn't usually a big deal when you're putting away leftovers that have been cooked, but sometimes it does make a difference. Like what if it's potato salad, and you're storing, warm potato salad in a deep dish like that? It's going to take longer to cool than something in a shallow dish over here. And potato salad is kind of notorious for sitting out at a barbecue, and it's got all of these starches in the potatoes and sugars in the mayonnaise that can feed bacteria. So you want to cool that stuff off quickly. All right, oxygen requirements. We can control microbes, depending on how much oxygen we give them and whether or not they need them. You totally need to know the names of all of these right here. So I'm going to take a screenshot again. It's going to be a very screenshot-driven class today. Okay, this term obligate. Obligate means needed, necessary. So obligate aerobes need oxygen. And I have that they need oxygen to transform energy in aerobic respiration. They need oxygen to make ATP, or to make energy. Make energy usually in the form of ATP or a proton motive force. Gotta have it. They don't have oxygen, they die. So die without oxygen. Whereas obligate anaerobes are poisoned by oxygen, so they don't use oxygen and they are poisoned by it. They make ATP with fermentation or anaerobic respiration, two processes that we'll be talking about later on in chapter 6. So obligate anaerobes don't use oxygen and are poisoned by oxygen. All right, now we're going to move down to this next to last bullet here down at aerotolerant anaerobes. Okay, they don't use oxygen, but they're not bothered by it. They're not poisoned by it. So they're aerotolerant, they don't care. They don't use oxygen, but if it's around, they don't care at all. It's, it's like if you don't eat gluten, you're probably not offended by the fact that someone else does, or that there's bread at a buffet table. You just don't pick it up. You don't need it. You don't care. It's not going to hurt you if you don't, you're not using it. Alrighty. There are microaerophiles that need oxygen, but only in tiny amounts. There really aren't nearly as many of those. The biggest class of bacteria that I think is really interesting are facultative anaerobes. If oxygen's around, they'll do cellular respiration. If no oxygen is around, they'll do fermentation. They don't care. They'll use oxygen or not use it. They're the most flexible microbes on the planet, probably. So an example of this is E. coli. E. coli is happy if there's oxygen around. If there's oxygen around, fine, it'll do cellular respiration. No oxygen around, no big whoop, it'll just do fermentation, and fermentation is plenty for making ATP if you're E. coli. Facultative anaerobes, they will grow with or without oxygen. Fun times. pH. All right, we already talked about how every protein, every cell is going to work hard at maintaining its optimal pH. For humans, it's near 7. And neurophiles, most human pathogens tend to be neurophiles, but there are some acid lovers out there, and those are acidophiles. H. pylori, this is Helicobacter pylori, is the bacterium that causes something like 90% of stomach ulcers. 90% of stomach ulcers are actually caused by a bacterium. And we didn't know that until like the mid to late 80s, I think, when some dude figured this out and ingested some H. pylori, gave himself stomach ulcers, and then used antibiotics to cure himself of those. Before that, people were just told, you just eat spicy food. You shouldn't be doing that. You're just stressed. That's why you have that. Well, stress definitely affects the body. Many cases, don't quote me on the 90%, but it's high like that. It's like 80 to 90% actually get better with a series of antibiotics if they are caused by H. pylori. Alkalophiles are microbes, are bacteria that like it slightly basic. Not too many of those, but there are some bacillus species that can handle that, partially due to their ability to make endospores. So let's see. So we talked about temperature, we've talked about pH, we've talked about oxygen availability. There is one more, water availability. And before you're like, what the heck is water availability? This relates to salt content of a microbial environment. So salt content. You know, from all that hypertonic, hypotonic, isotonic stuff that you had to learn in earlier biology classes, that if you put salt on a cell, water will exit the cell to try and dilute that out. That's called putting a cell in a hypertonic solution. Some bacterial cells can take that, some can't. Okay, the ones that can take a little bit of salt are osmotolerant. And an example of that is like a lot of staph species can take a lot of salt. Staph loves to grow on your skin. Your skin tends to be salty, especially where you sweat a lot. Halophiles like salt. They don't mind losing some water. They like salt. Alkalophiles, these guys are nuts. They love salt. They don't mind being dehydrated in the least. Oh, that noise that you hear is my dog jumping into her little sleeping basket. It was a basket that I used to keep magazines in until I got a dog, and she decided that magazines don't go in that basket. She goes in that basket. So that's what that noise was. Anywho, a lot of alkalophiles are actually archaea bacteria growing in these extreme environments like salt flats. It's crazy. All right, so water availability. All right, the last bit of this is that microbes will respond to what you put in the media, what you put in their broth. And a lot of them will have required elements. They need some sort of sugar to break down. They need something to make energy from. And I keep thinking that that PowerPoint here is actually something I can write on, and I can't. Not bad. Right, but growth factors like vitamins, a lot of bacteria, they don't care. Like required elements, this is some sort of sugar as a carbon source, carbon or energy source. Growth factors, vitamins and minerals. And microbes that require a lot of growth factors are called fastidious. Fastidious bacteria require a lot of growth factors added to their medium. So for example, Neisseria. Neisseria species tend to require a lot of growth factors. They're very fussy. Something like E. coli is not fastidious. E. coli will just grow anywhere under almost any conditions. It just doesn't care. Now this little bit here for energy and carbon sources, I used to have some other stuff written here, but basically what I want you to know the difference of I want to talk about things called autotrophs versus heterotrophs. Did I spell that? That E doesn't belong there. Heterotrophs. Autotrophs can basically make their own energy without carbon. So they don't require carbon compounds like sugars, as an energy source. So plants, which can take in CO2 and build their own sugars from it, they're autotrophs. They're actually photoautotrophs. Heterotrophs do require a carbon source as an energy source. Require let's say an organic compound. So for example, a sugar as an energy source. So just to talk about the difference between those two. There we go. Okay. There are different types of media that we can feed microbes. 99.999% of the time we feed them complex media, which is some sort of media where we don't know what the heck is in there. We don't know every single molecule that is in there. These are things like yeast extract. If we take some yeast and we hydrolyze it, we break up all those cells and take all the contents from those yeast cells and put them into a broth for our bacteria, we don't know every single molecule that went in there. There are going to be peptones in there, some small bits of protein, and it's just cheaper that way. Whereas the opposite of that would be chemically defined media, and that's where you start with distilled water and you put everything that goes in there, you know exactly what's going in there. You're putting this vitamin in and this sugar in and this, that, and the other thing in, but you know everything that goes in there. What a pain in the butt that is to make. Why do that? If you're just trying to grow some bacteria and keep it going, we feed them complex media. But if you're doing an experiment and you want to figure out what type of growth factors that a particular bacterium requires, you might do an experiment with chemically defined media to see where they and what they grow best in. Okay. There are times when we also need to know the number of bacterial. Oops, I jumped ahead. My bad. One more type of media, and you will know this for lab next week. Next week's lab, I think most of us are going to be doing an experiment on selective and differential media. We had to switch up. We had to move this particular experiment up so that our bacteria would not die. Selective media just enhances the growth of one particular type of bacterium and excludes others. So for example, this MacGonkey agar, MAC, inhibits the growth of everything but enteric gram-negative rods. Gram-negative rods, enteric means in your lower intestine, in your colon. So, I don't know. A former student called them poop bacteria. That's pretty accurate. There you go. Enterics. So For example, if someone has a UTI, 99, I don't know, some ridiculously high percentage of urinary tract infections are caused by enteric bacteria. So they would actually culture a sample of urine that was suspected of being infected. They would culture it on a MAC plate or something that enhances the growth of gram-negative rods and inhibits the growth of gram-positive. We also use differential media. Differential media makes different bacterial species look different. So for example, this MacGonkey agar, this MAC, okay, it's selective for gram-negative rods, but not all gram-negative rods are not the same. It'll make gram-negative rods that ferment lactose look pink, whereas those that don't ferment lactose will look just kind of creamy white. So pink means a lactose fermenter, or white means that it doesn't ferment lactose. Blood agar plates, or BAP, and this is another plate that we have with our kit that we'll be using, makes different streptococci look different. For example, the strep bacteria that causes strep throat will cause a clearing, or no, I don't want to use the word clearing, will cause the blood cells, that's not how I want to say it either. You get something called hemolysis of red blood cells, RBC, by Streptococcus pneumoniae. There we go. Whereas other streps don't lyse the blood cells as much. So MacGonkey selects for gram-negative rods, and it's also differential. So a lot of differential media is also selective. So make sure you know the difference between those. Alrighty, sometimes we need to get a count of how much bacteria is present in a particular sample. And there are different ways to do it. And this first way counts, the first method is by counting both dead and living cells. The easiest way to do that is using this Petroff-Hauser counting chamber. It's this thing down here. It's a slide with these grooves in them, like that, with a little cover slip on it. Right there. And what you do is you squirt your sample into one of these grooves and it gets sucked under the cover slip. And then you look at it under the microscope and you'll see a grid. And what you'll do is count all these bacterial cells and say, I didn't count them, and say that's 17, and it'll be like, well, that means that your original sample has 17 colony forming units per milliliter or something like that. And we would say, ooh, if this is a water sample, I wouldn't recommend drinking that. But the deal with Petroff-Hauser counting chambers is that we don't know if we're looking at this bacterial cell, if it's dead or alive. So we're counting both here. It can be useful, it can be helpful, but it's not always the most helpful thing. Another way to count both living and dead cells is with a Coulter counter, which is just a machine. I mean, you squirt your sample into the machine right there, and the machine says, bingo, you have this many cells per milliliter. It's really easy, but it's also really expensive. So, I mean, a slide like this is like 15, 20 bucks tops. Probably less on Amazon, and you can get it in one day. We find it more useful when we're, for example, testing water or looking at whether or not someone has a urinary tract infection, to count only the number of living cells in a sample. So one way to do that is with a serial dilution, and that is if we're talking about a sample that has a lot of bacterial cells. Like, let's say this right here. Let's say this tube right here is a urine sample. Someone had someone pee in a cup. right? If we took 1 milliliter of this out and put it on a plate and even did a streak plate, we would not be able to count individual colonies at all. It would just be a big smear of cells. So what we do instead is dilute it. We take 1 milliliter of this and put it into 9 milliliters of water. And the thing is, if we took a milliliter of this out and tried to plate it out, it would still be too much bacteria. So we dilute this again, and we take 1 milliliter of this and go into 9 milliliters of water. And then we dilute it again. And sometimes you get up to like a 10 to the 6, 1, 2, 3, 4, 5, 6 dilution. Then you can take a milliliter of that and put it on a plate or two. And then you'll have only, maybe 15 colonies to count. That's doable. If you have 150 colonies to count, that's going to be hard to count. 300 would be really hard to count. 600 impossible to count. So what you do is say, well, I counted 17 colonies on this 10 to the 6th dilution. So I have 17 times 10 to the 6th colony forming units per milliliter in that original. I'm honestly not sure 100% that we are doing a simulation of this in lab. We always do it in person, but I think I'm not sure if there's a simulation for this. I was playing online today, going through things, and I didn't see this one, so I'm not 100% sure, but it's messy and it's a lot of work. It's a lot of work, but it's helpful and useful because then only living cells are going to grow and form a colony. Now, what if you have a sample where there isn't a whole lot of bacteria? It's not very concentrated. There's just a tiny little bit. Like, for example, you know that the water you drink out of the tap is not sterile, but it's been tested to make sure that it has below a threshold concentration of bacteria. In fact, that makes me thirsty. I'm going to take a drink right now. Let everybody catch their bearings. For that, we use membrane filtration. So this is what we use if bacteria are not highly concentrated, and a lot of municipalities at their water treatment plants, they will test water using membrane filtration because you don't expect there to be a whole lot of bacteria in your drinking water, but you want to make sure that it's at safe levels. So what you do is you have a funnel with a filter in it. And you pour your sample in here over the filter, and filter paper, like regular paper, actually has microscopic holes in it. And it will the holes are small enough to let the water through, but the holes are not big enough to let bacterial cells through. So the bacteria stick to the filter. Can you think about this now? If all the bacteria is trapped on this filter, does it follow? Does it make sense to you that the water that we collect will be sterile? It will. And actually, filtration is one method that a lot of people who go backpacking, for example, camping, and where they won't have water supplies, they will use a membrane filtration unit to filter their water. Like water that you get from a stream. Good Lord, don't ever drink water from a stream unless you've filtered it with a system that will remove bacteria and will remove endospores and Giardia cysts from it. Right? These things used to be so ridiculously expensive, but as the technology has gotten better, now there are these nice little systems that work really well that are portable. Okay, so let's get rid of the water here. What we do then, though, for water filter, to figure out how much bacteria was in that water sample, what we do next is that we take this filter and we just put it right on top of an agar plate. And wherever there is a cell, a colony, a viable cell, a colony will grow. And then we'll count those colonies there. And it looks like this. Here we go. So this plate below, in the lower right, this red thing here is the filter. And I'm pretty sure this is E. coli. This is actually a McGonkey plate that selects for gram-negative rods. And it also is different. So it selects for gram-negative rods, but it's differential in that lactose fermenters. Lactose fermenters look either pink, or in the case of E. coli, if they're big-time lactose fermenters, they actually turn a metallic color. And this looks very metallic to me. That's coliform bacteria there. And that's what that looks like. And what people will do is count up the number of bacterial cells that are on there. Yeah. Membrane filtrations. Biofilms. I am not going to talk about biofilms now because we already talked about them a little bit when we talked about the capsule and the slime layer. Talked about that. So we got that. They do resist removal in antibiotics really hard because of that slime layer that they form. Okie doke, so chapter 5 will be all about control of microbial growth and how we use various disinfectants as well as temperature, to control for microbial growth. So we will pick up there next time. I hope you have a great weekend. Or no, wait. This is for Monday and Tuesday. I hope you had a great weekend, and I will talk to you about this on Wednesday morning or Thursday morning. So be well. And oh, no, wait. Gosh, okay, I'm totally losing it. It's been quite a day. This is for Monday. We'll discuss this on Monday afternoon and or Tuesday morning. And the rest of the week, we'll talk about chapter 5. Be well and stay safe. All right, let's get into chapter 5, which is control of microbial growth. Now that we have some ideas about what they need to grow, we can use that information to try and control the growth. And oftentimes we want to stop them from growing. In the laboratory, we're trying to get them to grow, but in life, on our food, for example, we want them to not grow. So there are different types of control. Sometimes we do just want to stop them from growing, but we don't want to kill them. We just don't want them to make more of themselves. Sometimes, in the case of sterilizing surgical implements, let's say, we do want to kill them. We want to destroy them. And sometimes we just want to remove them. We just want to remove them and not kill them. So there are different types of control, and we use both physical and chemical methods to do it. But first, a bit of history. There are some important people in the history of controlling the growth of microbes, and Pasteur is a big one. Okay, he's the swan-necked flask dude that showed that microbes exist in the air, but he proposed that microorganisms cause disease, so germ theory. He espoused germ theory, that human disease can actually be caused by microorganisms. This guy, Semmelweis, hugely important. He was a physician, I want to say in Austria. I don't know, it was Austria or the Netherlands. And he was the first person to suggest that doctors wash their hands before working on a patient. The history there is that he noticed that he worked at a couple hospitals. And at one hospital, he found a very high rate of women dying within days after childbirth. So the maternity ward had a high death rate at this one hospital, but at the other hospital they didn't. You know what? I think I'm going to go right back to notability here. Oh, I can't do that if I took a screenshot. Here. Get rid of that. There we go. All right, so we noticed that one hospital that he worked at had a high death rate among women in the maternity ward. This one had a low death rate, and I forget what the infection is. I want to say it was a strep infection, not uncommon after childbirth. Low death rate in maternity. Could not figure out how or why this was happening, and he looked at a lot of different things and found that in this hospital, interns and attending physicians would go to the morgue. Did I spell that right? After not spelling asymmetry correctly earlier. They would go to the morgue. Okay, this is where the dead bodies were, where they would have to do autopsies and the like. They would go to the morgue directly to the maternity ward to deliver babies. And over here, they didn't go to the morgue right before. I don't know, maybe the morgue wasn't next door to the maternity ward, so it took longer to get there, so they got distracted. Didn't go to morgue prior to maternity. And so finally, and this is after years of trying to figure out what the heck was going on here, he merely asked physicians, came up with the idea that, okay, it has got to be something in here, in this step. People are picking something up in the morgue and bringing it to the maternity ward that's killing women. So he merely proposed that they wash their hands after leaving the morgue before going to the maternity ward. The guy was ridiculed, apparently. Like, you can go do a little bit of research on him. I think he actually lost his job, and he just I think he developed severe depression after that as well. But yeah, he was ridiculed for it, but people started washing their hands. And washing their hands turned out to be critical. I mean, can you imagine? Can you imagine not washing your hands before even doing your work in the laboratory? I can't imagine even a dentist not washing their hands before, they ask you to open your mouth and start working on it, let alone a physician going to deliver a baby. So, Semmelweis. We credit this physician for getting people to wash their hands before doing all kinds of critical things. All right, Lister. Lister was the first to use an antiseptic during surgery. Listerine is named after Lister. And this guy, Koch. I don't know how to pronounce that, if it's Koch or Koch, like the Koch brothers. Not my favorite. But Koch, the microbiologist, actually won a Nobel Prize for it says here, isolated and identified microbial pathogens. He linked anthrax to a microbe that we call Bacillus anthracis. Yeah, he was one of the leaders in developing microbiology as a science. He's the first person who started using agar in agar plates, and actually it was his wife's idea. That he was, when trying to grow bacteria on a solid medium, he used potato slices. Can you imagine how gross that would be after a couple of days? And his wife was preparing some sort of jelly or jam, and agar is similar to gelatin, except that it's a plant product. It's not an animal-based product. And she was saying, well, why don't you use some of this stuff? Like, this jam got too hard. Why don't you grow bacteria on that? And that's what we use now. But he won the Nobel Prize, actually, in medicine for, I don't know, maybe I don't know if it's medicine or what. But won the Nobel Prize for linking microbial pathogens to human disease. I think he also linked the mycobacterium that causes tuberculosis to tuberculosis as well. Okay, so a little bit of history there. You do need to know these people. Okay, a little bit of terminology, because we try to do different things with controlling bacterial growth. Sometimes we just want to stop them from growing. Sometimes we want to kill them. Sterilization, destruction of all life forms. We're talking like for surgery levels. But sometimes we don't have to get things quite that sterile. Commercial sterilization just refers to sterilizing things that can grow under common storage conditions. Like, your pipette might be sterile, but not if you store it at thus and such a temperature. Or this can of food is going to be sterile, but not if you store it, under 70 degrees C conditions for 5 weeks, that sort of thing. We talk about disinfecting. Disinfection is for non-living surfaces. So if you're disinfecting something, you're talking about like disinfecting your kitchen countertop, that sort of thing, not your skin. For living tissue, we use antiseptics. So an antiseptic is for living tissue. An antiseptic would be like a mouthwash would be an antiseptic, not a disinfection. They do different things. Antiseptics are developed to work on living tissue and not destroy the tissue. Disinfectants are meant to be used on non-living surfaces. So bleach would be a disinfectant. You do not use bleach on living tissues. Sorry, Mr. President, that was a really dumb thing to tell people to do. Do not use bleach on living tissue. It will destroy your living tissue. We use antiseptics on living tissue. So like a mouthwash would be an antiseptic. It is okay to touch your tissue. Bactine, for example, it's a brand of disinfectant that is good for your skin. That's safe to use for your skin. All right. So here's a suffix. Suffix is a partial word that goes at the end of a word, side or sidal. So something that is bactericidal kills bacteria. Something that is bacteriostatic just prevents their growth. So for example, actually a lot of mouthwashes are just bacteriostatic. They just stop the growth of bacteria that would, be caught between your teeth, not necessarily bactericidal. And this is a word here, pasteurization. You can probably guess where it came from. Louis Pasteur developed this. And if you've ever had milk in this country, it's probably been pasteurized milk. It is not sterilized milk. If you buy pasteurized milk from the grocery store, you open it up and you pour it out, it is not sterile. It's not going to stay sterile forever. If you buy a jug of milk and don't open it and leave it in your fridge for 45 weeks, when you open that up, it is going to smell gross. It is not sterile. What pasteurization is, it's where you heat the milk or some other substance, but we use it mostly for milk. Heat the milk to, I believe it's 72 degrees C for
a short period of time. That time varies. Okay, so 72 degrees C is not going to boil. You're not boiling the milk. You heat it to 72 degrees for just a short period of time, like 10 to 20 seconds sometimes. And that destroys most of the pathogens that the milk would have picked up, from the cow's udder when the cow was being milked, or cow, goat, sheep's milk, whatever. So it kills most bacteria and helps the milk just stay fresher longer. But it does not kill all. It kills most, not all. I cannot emphasize that enough, because every semester I get students who think that pasteurization sterilizes. No, it does not. It does not sterilize at all. Now, a lot of people feel, well, and it's true, if you heat proteins to 72 degrees, you're going to denature a bunch of them. And so it actually will denature some of the milk proteins. So pasteurization actually does change the texture and flavor of the milk. And a lot of people are like, I want to do raw milk. I want to drink milk that is not pasteurized. About 15, 20 years ago in Massachusetts, they finally passed legislation saying that, yes, you actually dairies can sell raw milk to people, but there is a certification process that allows you to do that. You have to meet certain safety standards at your dairy farm, for example, to ensure that the raw milk is not going to be contaminated with much in the way of pathogenic bacteria. And I mean, people have to sign off on this, that I understand this is raw milk, there's bacteria in there, blah, 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 it has not been pasteurized. But a lot of people prefer the flavor of raw milk. And yeah, I mean, it should be absolutely safe to consume. You're just going to want to drink it a little faster, and you're going to want to get it at a reputable dairy where you know that they are very scrupulous about keeping things clean and doing things by the book and in a safe way. A lot of cheeses require using raw milk, not pasteurized milk. That will actually affect the cheese as well. So it's a big deal, pasteurized versus raw milk. But even if milk is pasteurized, it is not sterilized. Right. All right, now, when we're trying to decide, how we want to control bacterial growth, we have to consider several things. We have to consider the setting. Are we trying to clean your kitchen, or are we trying to clean a toilet? Are we trying to clean your kitchen, or are we trying to clean a surgical room? Things need to have a different level of cleanliness and sterility in an operating room. Types of microorganism. If we're talking yeast, you're going to want to perhaps use a different antimicrobial than if it is gram-positive bacteria. We also have to consider what it is we are sterilizing. For example, we use a flame to sterilize our inoculating loop in the laboratory, because those laboratory inoculating loops are made of titanium. They're made of metal. So we can flame them. They actually turn bright red when they reach a certain temperature, so we know it's sterile, and then we let them cool and we use them to inoculate bacteria. We don't have those loops in our kits. We have plastic ones, and we cannot flame them. We cannot use fire to flame and kill bacteria that are on plastic things. We have to do other things to get those sterile to begin with. And that's why we bought them pre-sterilized. All right, so I'm going to go back to Notability for a sec. There are both physical means to destroy bacteria and chemical means, not just to destroy, but to control microbial growth. For physical, we can use heat, we can use filtration, We can use pH, we can use water availability. Chemical means there are many different classes of chemicals. Lots of different, and again, it depends on whether or not it's a disinfectant or an antiseptic. Right. One of the biggest means that we use to control microbial growth is through heat. Now, there's dry heat versus moist heat. Moist heat would be boiling. Dry heat would be baking things in an oven. All right, dry is not effective as moist heat. Yeah. Moist heat, boiling, pasteurization, and an autoclave, which I'll talk about in just a minute. Now, you know that moist heat does not destroy endospores. Doesn't destroy endospores. But there are times that dry heat are more effective than moist. I mean, you're not going to boil, you're going to get a different result if you boil your food than if you bake it in an oven, if you roast it. But also, when we're dealing in the laboratory with RNA, or someone wants to work with RNA, we bake the glassware in the oven because for some reason that dry heat does a better job at destroying RNase enzymes. So the dry heat destroys RNase enzymes that are on people's fingers that cling to the glassware, and an autoclave doesn't really take care of that. Okay, so moist heat, boiling, we know about boiling. Pasteurization, we know all about pasteurization. Autoclave, this is pressure cooking. This is high temperature and high pressure. If you go to boil something, if you boil water, you are not going to get that water hotter than 100 degrees, right? Because 100 degrees is the boiling point of water. It's not going to get higher than that unless you put it under pressure. Then you can drive the temps up much higher. Then you can drive the temps up to, 200 degrees C if you want. And we do that in an autoclave. It's a pressure cooker. So we autoclave things in the laboratory a lot. And this will actually destroy endospores. So an autoclave will destroy endospores. So the food industry uses high temperature and pressure to preserve a lot of things where there could be endospores around. All right. Oh gosh. This one more time. And just to talk about how heat, if you have a bacterial protein and, it looks like this, if you heat it usually to about 72 degrees, that's often enough to just open up those polypeptide chains and denature the protein. Denature the protein. It has lost its shape. So that's why pasteurization works pretty well. It denatures a lot of the proteins there. So for bacteria that can tolerate high temperatures, I would say that their proteins are probably more heat stable than the mesophiles. So I just wanted to point that out. Heat will denature proteins. All right, we can use membrane filtration. Oops, this isn't a screenshot, and here I am trying to write on it. All right, we already talked about earlier about removing microbes by filtration. Just so that I can write on this again. If your little membrane filter, it's not showing up. There we go. Right in here, that's your filter that traps bacteria. The water that comes through here is going to be sterile because you've removed the bacteria. But if you take that membrane off and it's got all these bacterial cells on it, you've trapped them all. You have removed them. These cells here are still alive, but you've removed them from the water after it's been filtered. So this is really great when you need to purify water, or you need to purify materials that can't go into the autoclave. There are some things that in the laboratory, a lot of soap solutions, that if you autoclave them, they would turn into like a thick shampoo, and that doesn't work so well for us, so we filter them instead. Right. Physical. Okay. We can use different types of radiation to sterilize, to destroy bacterial cells. So the big three are gamma rays. Oh, blue's not showing up there. Gamma rays, that's a gamma. X-rays, you know what those are. And UV light will damage DNA that is inside prokaryotic cells. They will damage your DNA, so we do not use, UV light on humans. Actually, there is a type of nail polish where people have to use UV light to dry it. I've never had that done before, but I think there's certain types of gel nail polishes. And so people would go in for a manicure and have this done, and the salon technician would use a UV light to dry the nail polish on their fingernails. And scientists started coming out saying, oh my God, please don't do that, because you're damaging the DNA in your skin there. That's what the sun does to us when you get too much UV exposure there, and you can get skin cancer from that. Not a great idea. I mean, if you don't have to. So they have been developing that certain type of nail polish that doesn't require UV light to get it to harden. But it damages your DNA, and I don't want to have my skin that's on my fingers to be exposed to more UV light than they need to. Okay, but high levels will sterilize, low levels will pasteurize. But you've probably heard about UV irradiated food. They're foods that we take and we expose them to some UV light, not in the presence of humans. And after it's done, we sell the food. There was a lot of big deals saying, there were urban legends out there with people saying that, UV irradiated food has radiation in it. It's bad for you. You're exposing yourself to radiation. No, that's absolutely ridiculous. You put the thing in a box or in a room, you turn on the UV lights, you turn them off, you take the food out, the UV is gone. So it's actually a very safe way. If you want to talk about environmental reasons not to do that, not to use X-rays, for example, but that is something else. But after you've had an X-ray, you are not radioactive. UV irradiated food is not radioactive. And there's actually, with coronavirus, there's been a lot of talk about installing UV lights into office buildings and schools so that when no one is there, you turn on the UV lights overnight. And then when you come in the morning, you turn them off before anyone goes in. We actually use this in laboratories a lot. Where people do tissue culture, where you're trying to culture human cells that are a lot harder to grow and keep alive in a petri dish than bacterial cells are. And things have to be really, really clean. You do not want to get those cultures contaminated at all. So we use UV lights, and when people leave for the night, you turn on those UV lights for several hours, and that sterilizes the area. Right, I'm mentioning microwaves over here. Microwaves actually destroy bacteria from the heat that they generate. Microwaves do not damage DNA. When microwaves came out, oh God, gosh, the first microwave was probably in 1979 or something like that. And people were saying, microwaves spew radiation at people. They damage your DNA. So I actually had some colleagues who worked on that years ago, and they tried to show that microwave energy could damage DNA, and they could not find any evidence at all. They even repeated, there was one group that found that it did, and other scientists repeated those experiments and could not get similar results at all. So it was a fluke. They also found that group was not using proper controls either, so their data was refuted. So microwaves, they only kill by the heat that they generate. They excite the water molecules that are in food. Those water molecules start moving faster. That generates heat, and that heat kills pathogens. Before we talk about these other methods of control, there is another physical oh, wait, I didn't want to do that. I wanted to go to notability. Here we are. Let's get a different color here. Another, don't do that. Okay, so we talked about heat, filtration. Oh, we will get to pH and water availability. I want to talk about hand washing. Hand washing, which we do a lot of, removes bacteria, removes microbes, not just bacteria, yeast, fungi, removes microbes from your hands. It doesn't kill microbes. The soap is an emulsion. Soap forms an emulsion around bacterial cells. And then you rinse them down the drain. You haven't killed bacteria, you've only removed them. And thank heavens that antibacterial soaps have finally gone by the wayside, because they were no more effective at removing bacteria from your hands than regular soaps, and they also had chemicals in there that actually can induce bacteria to become resistant to it. So plain soap, plain hand washing is really great. We're not talking sanitizer. That's actually a chemical means over here. But hand washing with just regular soap is very effective at removing microbes from your hand. But you're just sending them down the drain. You're not killing them. Okay, so other methods of control. Low temperatures. So we were talking about temperatures before, and I mentioned this before, that they don't sterilize. They just stop the growth. They slow them down. Things aren't going to last in the fridge forever. And in fact, we just store bacteria in the refrigerator and freezer sometimes. Desiccation, that is lyophilized or freeze drying, and you have some experience with that because that is how our bacterial cultures arrived. They were freeze dried, and that's called desiccation. It basically just dries them out without killing them. And we use that to store bacteria. This last one here, water availability, salts and sugars. Now I'm going to go back to notability. We use salts to basically preserve food. Like beef jerky. You add salt to the beef, and that salt will dehydrate not just the beef jerky, but also the bacterial cells that are on it. You remove enough water from the bacteria, they die. Beef jerky, there is a fish called salt cod, and it's usually around Easter time. Sometime in the spring, it's a traditional food in a lot of cultures and a lot of cuisines. Salt cods, I always called it bacala. That's what my family called it. And it's fish that has been preserved with a ton of salt, and you gotta soak it for a long time. You have to change the water to get all the salt out and basically rehydrate the fish. But it's a great way to preserve food. pH. We use pH to prepare foods as well. There's a food called ceviche. Well, I'm talking about food here. Ceviche is a food that's in a lot of Central American cuisines. Puerto Rican cuisine, I've seen it. And it's where you take white fish, like a white flesh fish or shrimp, for example, and you squirt a bunch of lime juice on it, which is very acidic. And the lime juice basically cooks the meat. It changes the texture of that fish protein, and it destroys bacterial cells because bacterial cells, let's say you've got a protein here that has a particular shape. You could add heat to it to denature it, but you also can use acid to denature the protein as well. So it destroys bacterial cells there by denaturing their proteins. So we use that to prepare food. Sauerkraut is another thing. Sauerkraut is made, actually, we don't add acid to the cabbage. We let the bacteria that's on the cabbage create the acid themselves. And that keeps other bacteria from growing that can't tolerate the acid until it's really the lactobacillus that's in there. Lactobacilli bacteria finally produce so much acid that they can't even take it anymore, and they croak. And your jar of sauerkraut that once was cabbage is basically sterile. That's a jar of sauerkraut. That is a really bad looking jar of sauerkraut there. All right, so salt. Oh, sugars. Yeah, we use sugars to preserve food as well. High concentration of that will function like a high concentration of salt. So jams, for example, and then we can them. So there's a double whammy there. Okay, and that's a good stopping point, and we will pick up next time with chemical means of control. I hope you have a great week, and we'll talk to you in class. Bye-bye.